Next question we have, there's a couple which come from relating to Stevie's presentation about meropenem and the consequences for use in HITH, given that stability data. And I would say that the opinion that Stevie gave is very well thought out, but actually does contrast what maybe others in particularly the UK would contend they require to do because of their need to follow the yellow cover document, which is a very important guidance document for them in that country. I'm going to throw this next question to Adam about, in his experience and what his approach would now be, knowing what he knows about meropenem stability, about when he thinks an appropriate time would be to use in hospital and the home.
Thanks, Jason. Look, I think it all depends on initial antibiotic selection. I think if you're struggling, with particularly the Enterobacterales or perhaps Pseudomonas, and there's no other suitable initial agent and it pushes you towards meropenem, I think there are a number of things to consider. Certainly, anecdotally, there's no sort of, as Stevie sort of pointed out, it's just a smattering of small little observational studies. There's certainly nothing giving us confidence from the literature. But I think certainly anecdotally, there have been successes for its use. I think it perhaps could be used better. It's got quite a large, it's quite a good safety profile, and people can sort of generally tolerate quite large doses. So I think, a TDM approach with, 12-hour continuous infusion dosing, making sure patients are well educated, sitting in cool rooms, making sure that they've got their ice packs ready, and perhaps using, antibiotic level monitoring during the weeks, at least initially, to make sure that there's good drug exposure. Perhaps all of those things could be used, as a part of a good sort of OPAT HITH service in general, to sort of give you confidence about what you're doing. But I must admit, the way some, and I suppose selecting the right patient for that approach as well always comes into it, making sure that they're reliable and sort of the receipt of education is good. So those are the sort of things, and then we just have to wait, I suppose, until we get sort of more impressive data so that we can make better decisions on a larger scale with this. But at the moment, I would say it's more sort of, right patient, run out of other options and sort of, doing all of those little things. Thanks, Jason.
Great, Adam. If I could just chip in my opinion for what it's worth. I think a lot of these things depend on your risk appetite, and the risk associated with using it is pretty low, because the main concern you have is failure from very low drug concentration, which I think is unlikely given what we've seen about the stability data. But where there is a bit of uncertainty is whether or not the degradation products of meropenem actually elicit some sort of allergic response and cause cutaneous reactions. This has been reported in at least one study that I've seen, but isn't at all a common manifestation that people report as a common clinical observation. And so it's really weighing up those sort of issues that I would say is what a local ID practitioner would have to weigh up about whether or not they'd like to use meropenem in HITH. And sometimes there's not really any other option as well. So thanks, Karen Lim, for that question. And then I think that we've also responded to the question about whether there is sufficient evidence to give 2 by 12-hour infusions today in a day as opposed to a 24-hour infusion. And the answer to that would be that there's stronger stability data for that, but then there's the practicality and feasibility of whether or not you can have a nurse who's able to visit the home twice to reconnect the bag, or whether or not the patient can be trained sufficiently to do that connection themselves. Next question I'm going to go to is from Cara. And it's a question which she's directed towards Steve, which of course he's not here, so the panel will do their best to comment on this, about meropenem OPAT compared to intermittent at-home dosing. She comes from a service whereby they can administer TVS meropenem across a 12-hour shift, and so it's not truly 8-hour dosing that she acknowledges, but that is their approach to try and overcome stability by having that extra visit from the staff. And she says that her thoughts would be that having a continuous infusion of meropenem stable for 48 to 72 hours would deliver a better MIC profile than the intermittent dosing that we currently do. So I'll just firstly comment on that there's a number of simulation studies which talk about continuous infusion of a beta-lactam resulting in more reliable therapeutic concentrations than intermittent dosing. But this is done really, these data come very much from the context of high burden infections, really in the acute phase of treatment. And so whilst you're likely to get more reliable, higher concentrations with continuous infusion at any stage of treatment, usually by the time a patient has been transitioned into the home scenario, their level of sickness severity is greatly reduced. And so whether or not we need that same significant concentration of drug is uncertain. And so that's something which, not in the case of meropenem, is actually one of the sub-studies that's going to be looked at in the SNAP adaptive platform trial being led by Steve Tong and Josh Davis as part of the PKPD sub-study, which is being led by Amanda Gui and myself. And so, because I think that issue about what exposure of drug you should be looking for in the ongoing phase of treatment, the chronic phase of treatment, as opposed to the acute phase of treatment, is something which is very interesting. So, Cara, you have some other questions there. I will pass on your details to Steve and see whether or not we can get him to comment on those issues about probenecid and potentially increasing the half-life sufficiently to allow a change from 8-hour to 12-hour dosing. Certainly an area of current interest in New Zealand to use a lot of probenecid, particularly in Christchurch. We have reached the end of today. We've got a fantastic question here from Horst Field from New South Wales, which unfortunately we're not going to get to. But Horst, with your permission, I'm going to note this down and then work with Stevie to send a response to you. If there are others that are interested in this meropenem question, if you could just send an email to the CRE Reduce email address, then we'll also share this question as well as the solution or the discussion that we have. They're not called a solution, given we're talking about antibiotic solutions for administration in OPAT, possible in the home settings. So with that, I am going to thank everyone for joining today. We had a fantastic, generous audience here in the room here at the Royal Brisbane Hospital Education Centre. We had very good online participation, and I'm grateful to all of you for that. I would like to once again thank our sponsors for their fantastic support of our education series that we have at CRE Respond. I'm very grateful for the support that they do provide. Without that support, we are unable to provide the level of support and education that we are able to do. And so thanks very much to Pfizer, Gilead, Baxter, MSD, and our partners, the Hospital University in France, for their support. I would ask those of you that are still with us, if possible, could you please click on this QR code and answer the survey questions that are in there. We're always very interested in ways that we can improve upon the topics that we are providing to make sure that it's fit for purpose with what your needs as clinical practitioners and academics who are active in this space. And so I'd be very grateful for those of you that may be able to provide us a response to this so that we can continue to tailor our education to meet your needs. Our next seminar, which we are holding, is going to be in a few weeks' time, the 23rd of March, and is specifically focused at dosing in the Asia Pacific region, where we have some excellent speakers from various countries throughout Southeast Asia who are going to be presenting virtually, whilst we will have people gathering here at the RBWH Education Centre for a light dinner at 5:30 p.m. before a 6 p.m. start, Brisbane time, of course. So I would like to invite those of you that are able to come along to that to do so, but otherwise to register and join by Zoom. And then after that, on the 27th of April, we have our annual precision dosing seminar, which is going to be hybrid again. We're trying to cover all the continents of the world with this presentation, so we've had to divide this into 2 separate sessions. And you'll see the times there, 9 to 11 Australian Eastern Standard Time for session 1, which includes the US, and then 5 to 7 Australian Eastern Standard Time to include the Europeans as well. So that's another really exciting program, which I encourage you all to go to the CRE Respond website to have a look at, to register and to participate in as well. So thanks very much to you all again for joining us. We're very pleased to get underway this year with this education program. I'm really grateful to the speakers for their excellent presentations, to Eliza for her co-chairing, and remind you all that all of these presentations from tonight, as well as our previous presentations, which is well over 100, are all available on our YouTube channel. And so just go onto YouTube and type in CRE Respond, and you can find all of those different presentations there. Don't forget to follow us on Twitter and LinkedIn as well. So thanks everyone. Have a fantastic evening or day wherever you are in the world, and I look forward to speaking with you next time. Bye now.